in the particular field of research for which the Nobel Prize was given this year. So yesterday we had 3 lectures on physics, one on physics, one on Peace Prize, another on economics. Today we have invited 2 distinguished speakers to speak on Nobel Prize in chemistry and Nobel Prize in physiology and medicine. Friend Dr. Soumitra Das, he will be speaking on the hepatitis C virus, the research contributions from the Nobel Laureates. Warm welcome, Dr. Soumitra Das, for sparing your time and be a part of this webinar. Deliver the before that, Soumitra Das, I have 1 minute to tell a few sentences about your introduction, and in brief, I will tell that Dr. Soumitra Das, 5 years at University of Science as visiting professor. Both of us jointly coordinated 4 or 5 refresher programs in biology. And I learned many things from you, sir. It is a great time for me. And at present, Dr. Soumitra Das is the director of National Institute of Biomedical Genomics, Kalyani. He is a microbiologist. And before Kalyani, he was microbiology and cell biology of the Indian Institute of Science, Bangalore. He is known for his studies in the field of molecular virology and molecular biology. Professor Das is a distinguished elected fellow of all the 3 science academies of the country, like Indian Academy of Sciences, National Academy of Sciences, and Indian National Science Academy. The Department of Biotechnology of the Government of India has awarded him the National Bioscience Award, Career Award Development, one of the highest in the Indian Science Awards, for his contributions to biosciences. Professor Das is focused on translation of hepatitis C virus. The team of translation of a special hepatitis C virus. The research conducted by Dr. Anup Korande of the Department of Biochemistry at IISc has resulted in successful development of a vaccine for hepatitis C, for which the Nobel Prize is given for their other contributions. So we are extremely fortunate, Professor Das, for having you here. And Professor Das has many string of awards. I will not get into this, but he's a distinguished colleague and a distinguished scientist. It is my pleasure. Welcome, sir. And now, technology and medicine. Dr. Soumitra Das.
Okay.
Professor Ayyappan has joined he's the chairman of the academy, has joined. I extend a very warm welcome to you. Yeah.
Professor Ranganath, introducing the welcoming and Rao. And Sir Rao, welcome to you, and request your talks.
All right. Thank you for your nice introduction and the welcome. Actually, it's a real pleasure. Can you hear me now?
Yes, sir. Yes, sir. Yeah. Please go ahead.
Okay.
You can hear.
So, yeah, it's a real pleasure to join you people for celebrating the science. You know, it's the first time that I'm actually giving any lecture in the Karnataka State Science and Technology Academy. Thanks to Professor Ayyappan and thanks to Professor Ranganath, my longtime friends, and the other organizers for this Nobel lectures. You know, as Professor Ranganath mentioned, that our lab in the Indian Institute of Science have been working on hepatitis C for last 22 years. And it so happened I'm fortunate to work with this particular organism since its inception. I think it was discovered in '89, and then I started working on this virus in '91. When I was in UCLA, and then after I came back to India, joined Indian Institute of Science, and my lab started working on hepatitis C virus in different aspects. Currently, as he has mentioned, that I am on deputation at the National Institute of Biomedical Genomics in Kalyani, but my laboratory is still there in IASC with a full group working in my laboratory. So I always say that my head is here and the heart is there. So, I mean, and I'm actually a Bangalorean now, I think, being there in Karnataka for 22 years, and I think the rest of my life. So it's a real pleasure, and thank you for giving me this opportunity to present. Now, as I said, that hepatitis, this whole concept is actually, if you really look at the historical perspective, it goes back to 3000 BC, where the Sumerians, they used to call that liver is the home of the soul, and jaundice is the attack on the liver by a devil named Azau. And in 400 BC, Hippocrates mentioned that the bile contained in the liver is full of flame and blood, and it erupts. After such an eruption, the patient soon raves, becomes angry, and talks nonsense. Then between 17th and 19th century, people started talking about the jaundice epidemic in military men and the civilians. Actually, there are common in the French camp, they used to call yellowing of the camps or the battlefield jaundice. They call the jaundice de champ. And then icterus was the clinical term. Actually, this icterus is Icterus galbula, which is actually a Baltimore Oriole. And the myth was that sighting this bird was supposed to cure a person with jaundice. So people were actually throwing the ideas of this, how this disease is originated, and that's the time in 1965, Dr. Bloomberg was actually trying to study the genetics of disease susceptibility. And he accidentally ended up discovering with an antigen, which is actually causing this hepatitis. And it was named at the time as an Australian antigen. Now, hepatitis for everybody, we know that can be triggered by different things. It can be triggered by alcohol. It can be triggered by autoimmune disorders. There is non-alcoholic steatohepatitis. Many a time it is because of the toxins and other infections, but more so is because of a range of viruses, which we actually designate with different alphabets. A, B, C, D, E. I always call that F could not sustain, it failed. So F is not there. After that, it becomes hepatitis G virus. And there are some non-hepatotropic viruses also that can cause viral hepatitis. Now, if you really look at this A, B, C, D, E, and G, all these viruses are actually, most of them are single-stranded RNA viruses, except the hepatitis B, which is a double-stranded DNA virus. But if you really look at it, the extent of infection is highest so far is by the hepatitis B, which is 240 million chronic infections globally. Then 150 million is chronic infections globally for the hepatitis C. And, as I was mentioning, that 1989 is the time when hepatitis C was actually noticed that it causes the liver cirrhosis that leads to hepatocellular carcinoma. And in 2007, there is an estimate that it actually surpasses the HIV. So there was incidence of 15,106 compared to 12,734 in a particular zone. So this hepatitis actually in 15 to 25% cases can be eliminated by our host immune surveillance or actually host protection. But 75 to 85% cases, it can establish a chronic infection, which leads to chronic hepatitis. And 20 to 30% of that can lead to cirrhotic liver, which we call cirrhosis. And 2 to 7% of that population can actually have the hepatocellular carcinoma, which is end-stage liver disease. Now, how people get hepatitis C is mostly by injecting the illicit drug use, which is around 53%. There is high-risk behavior in the past. There are 10% for sexual activity. Blood transfusion, actually, a lot of people got it during blood donations and blood transfusions. Actually, before 2000, hepatitis C testing was not mandatory. So a lot of people actually occurred because of the blood transfusion. And also some of the accidental cases of healthcare employees getting the hepatitis because of the needle prick and other things. Now, if you look at the hepatitis C, it's actually prevalent now all over the world. And there are several genotypes. There are 7 different genotypes. And there are 62 subtypes. And, it's keep on changing because the virus keep on mutating. Because of the faulty RNA-dependent RNA polymerase, it accumulates mutations and it actually changes its genome frequently. And that's how the challenge is. And as I told you, it's a long journey. And I'm going to tell you how actually these 3 people who got the Nobel this year actually somehow revealed the mystery and actually unfolded the mystery of hepatitis C and finally laid the path towards a recovery from this infection. Now, these are the 3 people. So I'm going to highlight their discovery to the treatment. And the 3 people that we are today celebrating are Harvey J. Alter, Michael Houghton, and Charles M. Rice, whom I actually work closely. Now, what actually Alter's contribution is, when hepatitis B was actually discovered, then there are still some cases where people used to see that there is similar kind of symptom, but it's not quite hepatitis B or hepatitis A. So they used to call it as a non-A, non-B viral hepatitis or NA-NBH. Now this actually was unfolded again. I mean, he investigated, Harvey Alter investigated and found out that actually if you take the serum from this kind of NA-NBH patient and try to inject it in chimpanzee, you can actually reproduce similar disease, similar symptoms in chimpanzee. And that was the first evidence to show that this chronic infection is other than something which we knew a long time. Hepatitis B was actually known from 76 onwards, when Bloomberg actually got the Nobel Prize for the discovery of hepatitis B. So non-A, non-B viral hepatitis was actually first time thought about by Harvey Alter. And he showed that serum from patients with acute or chronic NA-NBH could transmit the disease to chimpanzee. And not only that, they also pointed out that it looks like the virus is coming out or coated with a lipid. And so the lipid droplets are actually being used for the virus transportation from the cells to other cells. And this came out in 1978 Lancet. And that was the first time it was noticed that there was another virus which can cause a viral hepatitis other than A and B. So immediately people thought that this could be the hepatitis C. Then comes the contribution of Michael Houghton, who actually identified the infectious agent as hepatitis C. How did they do it? So they have actually taken the plasma of the infected chimpanzee. Infected means they were infected from the non-A, non-B viral hepatitis. And that plasma, they pelleted down, isolated DNA and RNA from there, and had a shotgun. At that time, sequencing was not that easy. So they made a cDNA library in lambda GT11. And that cDNA library was actually probed with the antibody, serum antibody, isolated from the patient who has developed non-A, non-B viral hepatitis. So you have now the nucleic acids which can make their protein in the lambda ZT11. It was expressed, the protein were expressed, coated, and then it was probed with an antibody to identify, do you see any protein which are correspondingly neutralize these antibodies? And after screening several clones, they found out one clone called 511, which actually showed that it is extrachromosomal, derived from an RNA around 9,600 nucleotide, and only found in the non-A, non-B viral hepatitis samples, and distantly related flavivirus. That's how. Remember, this we are talking about 1989. It came out in Science by Chu et al.'s paper from Michael Houghton's group, and they actually pointed out that this virus encodes the protein which can bind to the antibodies that are found in the non-A, non-B viral hepatitis. And this is essentially the Western that was actually shown in the paper, showing that the non-A, non-B viral hepatitis are lighted up with the antibodies collected from the NA and BH patient, but not from hepatitis B, not from hepatitis C. So that was actually the identification of the infectious agent for the non-A, non-B viral hepatitis as hepatitis C virus. Now, Charlie Rice comes into the play, and he tried to further characterize these things and move it forward. Establishment of an efficient infection model for the hepatitis C. So what he did, he has isolated and cloned the virus from an infected patient by a reverse genetics. And then he actually identified a conserved non-coding region at the 3' end, which actually is very important to be in the genome so that the genome can replicate. And so he has isolated the virus from the infected person, cloned it, engineered the RNA, and showed that there is a conserved 3' UTR. And if you use this infectious RNA transcribed in vitro, infect it to chimpanzee, you can actually reproduce the viral infection. So that was actually a breakthrough, because before that, we didn't have any handle on the infectious cDNA for the virus. And as you know, for molecular biology, you can't do much unless you have a cell culture in hand. So he also showed that now if you isolate the virus from infected chimpanzee, you can infect into a cell culture. He named it as HUH7.5, and that cell culture, it's a human hepatocellular carcinoma cells, and that cell culture could successfully replicate this viral RNA. So this is, again, this is now 1997, published in Science by Kollikov et al. From Charlie Rice's group. And this is definitely now the discovery part of the hepatitis C was complete. So as I told you, it started with a blood transfusion from the discovery of the non-A, non-B viral hepatitis. The serum was injected into chimpanzee, and this was actually then probed with the serum of the antibodies collected from the infected patient. The genome was isolated, genome was injected, viral RNA genome was injected into chimpanzee. The infection was reproduced, isolated the serum, and then isolated the serum-derived virus, infected in a cell culture, and now the infectious cell culture model is established. And that is, I think, 1997 and 1998, when I joined in the institute, I was lucky to have the clone and the cell culture from Charlie Rice's group itself, and try to see how we can start in our lab. Now, this is a significant contribution because that helped us or the whole world to develop different sensitive hepatitis C virus tests for the detection and the elimination of the risk of transfusion-associated hepatitis. As I told you, before 2000 in India, the blood donors were not mandatory to check hepatitis C presence. And there are unfortunate cases where after 17 years of the blood transfusion, people came to know that they are actually carrying the hepatitis C virus. So it takes long time to actually manifest the disease, but that it was actually spreaded in the population unwantedly because there was no detection system in place. So this helped, and as you all know, ultimately last 5 years back, now this effective antiviral drugs are also available in the market. Now the tenofovir and sofosbuvir people are actually getting almost 98% clearance of the viruses. But so the disease can now be cured and raises potential for the global eradication of hepatitis. Actually hepatitis C researchers, we usually meet every year. We call hepatitis C virus meeting. So I think 2 years back, I was also there. Charlie Rice was mentioning that probably now 2020, we don't have to have a hepatitis C meeting. Now it's so coincidental, we could not have the meeting. That's not for the hepatitis C is eradicated, but because of the COVID, we couldn't have it. And Charlie Rice got the Nobel along with Harvey Alter and so it's a big boost to the scientific community, that how you can actually start from the beginning and then take it to the cure or take it to the treatment of the disease. So discover the disease and make a roadmap to how to eradicate or how to actually treat the disease. Now I will just switch gear a little bit and tell you a little bit about our research as well and the hepatitis C virus, how hepatitis C virus prefers to work or infect only liver and not in other organs. Now, this is an RNA virus, positive strand RNA virus. The viral RNA goes inside the cells. Virus uncoats the viral RNA, and that RNA is used for translating to make the viral proteins. And these viral proteins are then used, some of them are actually used to make the replication of the negative strand. Now, from the positive strand, negative strand is made, and this negative strand is used as a template to make more and more and more of positive strand, because you need 100 times more positive strand, because that is actually ultimately getting packaged in the virion, which we call virion assembly, and actually egresses out or releases out and affects the neighboring cells. So this is a simple life cycle. I actually oversimplified it. There is a membranous wave structure near the endoplasmic reticulum where this whole assembly happens, the translation and replication, and then finally it goes out in the lipid pathway. Now, as I told you, that hepatitis C prefers liver. So if you really look at the liver, there are several cells, and hepatocyte is one of them. Now, hepatocytes are the ones that are infected by the hepatitis C virus. But if you really look at the neighboring cells like stellate cells, Kupffer cells, they don't really get the virus. So there is a possibility that something is there in the hepatocytes which occupies 80% of the cells in the liver, but not the endothelial cells, stellate cells, or Kupffer cells, which are actually target of the hepatitis C virus. So we asked the question, why? This is the same time, I will say, 1998, came back to India and joined Indian Institute of Science, Department of Microbiology and Cell Biology. We started addressing this question. And, to me, it looks like somebody is getting into a house and they exactly know where to go. They find out the kitchen and they go and make use of those kitchen vesicles, all these vessels or the spices, so that they can make palatable food. So they know precisely if they ended up with the neighboring rooms or other places, they may not have an access to the vessels and the oven and other things to make palatable food. They indeed make good food, and this food is actually the one which we call a viral polyprotein. So viral RNA gets translated, and then the viral cellular proteases and the viral proteases clips it, chops it like the way we do the sub sandwich into small pieces. So the core, envelope, and the P7, the ion channel protein, these structural proteins are separated out, and the non-structural protein, which are mostly functional, are separated out. Of this non-structural protein, there is two protein I wanted to attract your attention. One is the NS3 protease, which is being targeted for designing antiviral, and the other one is the NS5B, which is RNA-dependent RNA polymerase. And the third one is the NS5A, which is also notorious because it contributes mostly in the pathogenesis. What it does is actually it inactivates our body resistance by interfering with the interferon pathway. And so arguably, you can see that earlier, although the hepatitis C virus was actually treated with pegylated interferon and ribavirin, which is a nucleoside analog, but the success was only 40%. Most of the patient did not respond because of this NS5A was actually inactivating the interferon path. But now, because of the discovery of this Charlie Rice and the others, now you have a cell culture system. You can make the viral protease. You can do the replicon assays with Wattenslager in Germany. He also contributed quite heavily into this, and several other scientists. And they showed that if you can actually target this NS3 protease with telaprevir, you can inhibit the replication of the virus, or the sofosbuvir, which actually targets the NS5B RNA-dependent RNA polymerase. You can actually bring down the virus to almost non-detectable level. And lately now NS5A, ladybasfid, is also being tested to inactivate the NS5A and then inhibit the viral replication. So, but you know, the drugs are in place, but the problem is not really over. As you know, that I told you there are several genotypes and there are several subtypes of hepatitis C. And you know, emergence of drug resistance is always keep going on with these kind of RNA virus mutations. And relapse is another thing once the drug is stopped. There are several cases after 6 months, the patients are coming back with the hepatocellular carcinoma, or there is a relapse of the disease. So, and additionally, therapy associated side effects are also there. So people try to find out, is there any other way so that we can find out a better treatment option, or actually we can have an additive treatment option so that we can somehow prevent the progression of the disease. See, bringing down virus titer is one thing, but to inhibit the progression of that disease is another challenge. So, as I was referring that when I joined in 1998 in this institute, IISc, so we asked the question that, you know, what is actually unique about this virus that it causes such a havoc pathogenesis? And when we looked at the molecular biology, we found that all the cellular RNA or the host mRNA, they are capped at the 5' end, where the cap binding protein binds, then other initiation factor binds, then ultimately the tRNA comes with a battery of initiation factors. With the 40S, they scans for the initiator AUG and starts the 60S joins and the starts protein synthesis. This we all read it in the biochemistry book since class 10. But this virus is little different, as I told you. It does not have the cap structure at the 5' end. Instead, they have a very structured RNA element, which we call internal ribosome entry site, which is a scaffold of RNA that actually gives a place for the 40S to land on near the initiator AUG. So, virus in this viral RNA, the 40S does not bind to the end of the RNA, but it binds to this scaffold of RNA structure near the initiator AUG, and then the 60S joins and starts translation. This internal initiation of translation makes it very, very different from the cellular RNA. So what we asked the question in that time is, now if you look at the ribosome, this is the head, this is the platform, this is the beak, this is the front gate where you have the A sites and B sites, where the viral RNA makes a contact. I mean, cellular RNA makes a contact. But the hepatitis C virus, such a clever way evolved that it doesn't really bother with the cellular front gate. It uses the back gate, triggers a conformational alteration and access to the front gate to synthesize their protein. So when we established the lab and the research group, our question was, is it possible that we find out that how viral RNA bribes the ribosome, cellular ribosome, so that they can make contact with the backgate man and then get into the ribosome. What are the host factors they are actually manipulating, and what are the viral proteins that are actually involved in regulating the viral RNA translation. That's how we have been working. The reason behind it, since it is unique and fundamentally different, is it possible that we can understand the biology better, and can we really target it to design antiviral? And we were actually successful to some extent. One of the small RNA that we showed, which is part of the viral 5' untranslated region, my first student showed that if you use that RNA binds to the ribosomal protein S5, which is actually sitting at this exit gate, which the viral RNA is actually bribing. Now, if this small RNA binds to this exit gate, the viral RNA cannot access to the exit gate. It cannot bind to the 40S ribosome. The translation is stopped. This is actually, I think we started in 2004, we published it. We have been still doing it, characterizing this RNA. We're now trying to, this is a US patented RNA, which we are trying to project it as an antiviral and try to see it can be a selective antiviral. Another thing that we have actually, I want to bring your notice, is this: there are several host factors the virus somehow manipulates. Now, these host factors are actually nuclear resident. They are nuclear proteins. They have functions in the nucleus. But in the infected cells, there's some kind of a hijacking situation. They are forced to come out from the nucleus to the cytoplasm, bind to the viral RNA, and helps in viral RNA translation or replication. One such protein our lab have been working for a long time, which is human La protein or La autoantigen. And we showed that the La protein actually binds. And if you just take the RNA binding motif of that La protein, which is actually a beta hairpin structure, which we solved the structure in collaborations with Dr. Siddhartha Roy at IICB Kolkata at that time. And we showed that if you use this 6-7-mark peptide, this peptide is good enough to block the binding of the full-length protein and inhibit the viral replication. Not only that, we also showed that if you add 6 arginine residues to make it more hydrophobic, it goes inside the cells and then goes to the liver cells and inhibits the viral RNA like a surgical strike. So this is something very interesting. We actually now, again, US patented it, and we try to cyclize this peptide, make it more stable, and now trying to commercialize it to see whether we can actually stop the viral RNA and the ribosome interaction. We all are familiar with the bacterial RNA and the ribosome interaction inhibition. There are several antibiotics there, but now this is the same concept we are extrapolating to the viral RNA and the ribosome interaction, and can it be interfered with the small RNA or a peptide? And that was actually our goal. Another thing was bothering. Remember, I was just saying that it infects chimpanzee, but it does not infect many other animals. Something like, if you really look at the mice, rabbit, or even other rodents or primates, it doesn't infect any out there. So is that possible that something is missing there? We did show that between human, chimpanzee, mouse, and rabbit, if you look at this human La protein, we have a conserved sequence in human and in chimpanzee, which are actually permissible to this viral infection, but not in mouse, not in rabbit. So is it possible now if you mutate it back and restore it? And that's how we did. We showed that the mouse La does not bind to the viral RNA, but when you mutate it back or humanize it, then that La can actually promote translation. When we did this experiment in 2014, CRISPR-Cas was not common. Actually now, DNA is going to talk about the CRISPR-Cas. That technology is available now. We are trying to knock out this mouse La and knock in the humanized La, and then try to see whether we can have a mice model, which is still pending. Although now we feel that the hepatitis C, everything is solved, but this is still, we don't have a lot of answers there, and we don't have an animal model. We are actually working on it and see whether we can have an animal model. As I was referring, that whole translation, although looks very simple, but it's actually nicely orchestrated by a nuclear to cytoplasmic relocalization of the proteins. There are several proteins, as I told you, like La, HUR, PTB. They are all nuclear resident. They have their function in the nucleus. But the infected cells, they are brought out to the cytoplasm. They bind to the viral RNA 5' UTR and 3' UTR, and this is essentially the confocal showing their migration pattern. And then they bind to the viral RNA to help in translation or in replication. But what is actually different between the nuclear protein and the cytoplasmic protein is actually, we are now addressing it. We hypothesize that there are modifications, and that's how they come out. They lose their visa and they come out. And because of the modification, they have altered affinity. And of course, the availability is also increased because of the nuclear to cytoplasmic relocalization. And that's essentially the change again. So essentially what we can see that in the liver cells, there is a continuous tug of war between the host cell and the virus. And we have shown there are proteins like HUR. There is another microRNA called MIR125B. They actually keep on tilting the weight towards the virus. And then they found that if there is a La protein, which we call now OLA, then actually the whole thing is towards the virus and the host cell gets defeated. But this is also now regulated by a traffic signal. And this traffic signal is the modifications that we are actually trying to understand, which will decide the abundance of the protein in the cytoplasm or in the nucleus. So largely, this is essentially an orchestra where all these proteins, RNA, and the small RNA, microRNA, or the long non-coding RNA are doing their role, and HCV is actually somehow tuning the whole liver to dance with them. And that's how we are actually trying to study over the years. And we showed which all proteins are involved, which are long non-coding RNA or microRNAs are involved for this replication and the translation. Last couple of slides, I'll just show you quickly that we have recently worked with one more long non-coding RNA, which is actually abundant in the liver. And we showed that this RNA, long non-coding RNA, helps the virus to load it into the lipid droplets and then transport it out from the cells. It's not only that it increases the size and the number of the lipid droplets, it also helps in the core protein of the HCV to be loaded onto the lipid droplets, and it helps how we can go out from using the VLDL pathway to affect the neighboring cells. But within that is proviral, but within the host also, they have another microRNA, which I did not really talk about today, but that microRNA actually somehow arrests the long non-coding RNA and keep it under check, and that's how the virus can establish chronicity in a particular liver cells. So we believe that we have solved to some extent the mechanism of establishing the chronicity in the liver, which we call the antiviral effect of the cells. So essentially, we have been studying the infection and trying to understand the viral protein and the host factors like microRNA, long non-coding RNA, and the protein. I told you, actually, these are the different things. In parallel, in the lab, Government of India asked us to see whether we can find out some herbal compounds which will be effective for preventing the hepatitis C spread. And we were successfully able to isolate two compounds, punicalagine and elachic acids and punicalin. These compounds are actually can inhibit the NS3 protease to some extent to the telaprevir which is available. And we actually found out the molecular basis. Another thing we showed that corylagine and phyllanthine, this is Phyllanthus amarus, which we call bhui amla. Tagrajan from University of Chennai have shown long back that this bhui amla is being used by a lot of people for viral hepatitis. Actually, he went even for human trial, but we didn't know the mechanism. Now we could actually decipher the mechanism. We could identify the compounds involved into this bhui amla, that is phyllanthine and corylagine, which can actually inhibit the NSC protease of the virus, because Srinivasan, my good friend at MBU in IISc, with him, we showed that actually it goes and sits in the catalytic groove of the enzyme and efficiently inhibit the viral protease. Why I'm showing this? Because these are natural compounds. It's actually, we believe, and this is very recent study with organic chemistry, we have modified the compound Phyllanthus with Jayaraman at the IISc organic chemistry, and we showed that actually we can make it even more effective for inhibiting this hepatitis C. And all these things we have tried in a chimeric mice, which is harboring human hepatocytes. As I told you, the regular mice does not really infected by the hepatitis C, but there is a chimeric mice available in Japan, which we have collaborated and done it, which harbors human hepatocytes. And in that system, we could show that if we use these compounds, isolated from Phyllanthus or Phyllanthus amarus, this gorilla gene, it can protect the viral infection to some extent. And more importantly, it actually prevent the collagen deposition and progression of the liver fibrosis. This is something very important, which we are talking, that this virus, that drugs that are available, tenofovir and sofosbuvir, can actually prevent that, bring down the virus titer. But we have shown that in addition, if you use this pomegranate Punica gene or gorilla gene from Phyllanthus amarus, we can actually restrict this progression to hepatocellular carcinoma. So this part is still unsolved. Another thing in parallel, we were trying to make some vaccine candidates. We made a virus-like protein particle, which we call VLP. This Professor MS Shaila started, and then with collaborations with Anjali Karande's lab, we have actually shown this VLP can elicit immune response in mice. We are now trying to do it in pig. The initial results are very, very encouraging. We are trying to now go into preclinical trial and try to see whether first time we can actually contribute to the hepatitis C virus vaccine. So I started the lab with a very minimal translation. The question was very simple, that how the proteins are synthesized, which we call translation, but ended up doing a lot of translational work, because all these observations we tried to translate it to make it antiviral, be it a small RNA or peptides or even monoclonal antibodies or compounds from different natural sources or a vaccine project. And all of them are being tested in a humanized mice system so that we can actually move. But this is all possible because of large number of collaborations that I do, and that is the strength of our lab. We work on antivirals, host-virus interaction, pathogenesis, liver mechanobiology. We work with mechanical engineering of Indian Institute of Science. Now we call BSSC, Biosystem Science and Engineering. I had actually students graduated from mechanical engineering background, addressing the liver mechanobiology under hepatitis C virus infection. We also work on liver-targeted drug delivery. And as I showed you, that we are trying to make the VLP and DNA vaccine as an Indo-Australian project. We also work on monoclonal antibody and neutralizing antibody isolated from infected patient. And the collaborators are here. But, you know, all these efforts are started, as I told you, my journey also started in 91 when I was in UCLA with hepatitis C virus, and still we are going on. But I can see there are several questions still remaining. Number one is the appropriate model organism. Chimpanzee research work is now heavily discouraged. Only non-human primates susceptible is at this moment is chimpanzee. So practical and ethical constraints are there. We are trying to develop mice model. Humanized mice and ectopic liver generation is the only option. Vaccine, as I told you, here also it's a big challenge because there is a variations in the viral RNA sequence because of the error-prone replications of the RNA-dependent RNA polymerase. There is extensive glycosylations of the envelope protein that makes it more resistant, and different epitopes are being masked, and limited production of the whole inactivated virus. This is something is a big challenge. Even for COVID, now we are actually trying to make large-scale inactivated virus, but that is still not possible for hepatitis C, we still can't make large amount of hepatitis C virus in the cell culture. So that is actually impeding the success of developing a vaccine. And as I mentioned repeatedly, that hepatocellular carcinoma progression is a big challenge. We know that the pathways that are activated because of the viral infection, unless they are stopped, then the progression to the liver carcinoma cannot be stopped. So we know the pathways, but we are trying to look for inhibitors of those so that we can stop the progression of the disease. There are epigenetic modulations of the RNA binding proteins of the nucleic acids, and that also contributes to the disease progression. We're trying to identify that. And there are triggering secretory molecules such as exosomes, and that actually carries the virus from liver to other organ. Now, at the latest stage of viral infection, it is moving to other organs. That is dangerous, and that is done by the exosomes. We are trying to understand the compositions of the exosomes, how they can actually be prevented from transporting from liver to other organs. So, the research continues. And, the last thing that I just heard recently, a message from Charlie himself, he says, let's hope that this example means like this, the journey of hepatitis C virus for him is of the benefit that science can bring and will encourage the public to embrace and support fundamental curiosity-driven research. This I'm going to harp on it. I think people have to be curious. They need to ask real questions, real good questions. And as we are in the midst of experiencing with the ongoing pandemic, there are many challenges ahead. I'm sure if we can actually put together our effort, we should be able to win over COVID also soon, and we should be able to be actually guarded ourselves in a way that in future we won't be really shaken up with another RNA virus infection. With that word, I would like to thank again the organizers for giving me this opportunity to share my thoughts with you. And it's always a pleasure to work with such an organism, which actually we could at least got some solution to it. And I'm very hopeful that we'll be able to get some solution for COVID-19 as well. Thank you.
For this enlightening talk of yours, and the whole the Nobel Physiology, medicine, the whole history behind it, and all that we wanted to know about hepatitis, you brought in these few minutes. And in spite of your busy schedule, moving from Bangalore to Calcutta, and a meeting this evening, in spite of that, you have kindly accepted our request. So very grateful to you from the Karnataka Science and Technology Academy. I would request Professor H. Ranganath for his words at this stage.
Just again, I reiterate my grateful thanks, sir, for sparing your time. And sir, I'm extremely grateful to your final message. The final message from one of the Nobel Laureates, no? Curiosity-driven research. And I add one more thing, curiosity-driven teaching, followed by curiosity-driven research. If both things can happen, our science can progress. And another message from you is extensive collaboration. Extensive collaboration to address one problem. That extensive collaboration has given you a lot of results. And sir, within 40 minutes, starting from the discovery of hepatitis C, and you have got to the extent of finding a vaccine, it's a great successful story. And all of us are benefited of our expertise. Talk, sir, on behalf of the Academy and on my own behalf, I'm extremely grateful to you, sir. Thank you very much, sir. Thank you. Thank you.
Thank you. Thank you.
We have the next Professor D.N. Rao, esteemed colleague from the Institute of Science. Again, from the Academy, so we are so grateful for accepting our request. Professor D.N. Rao would speak on the for 2020 in the subject of chemistry, and I would request Professor Ranganath to kindly introduce Professor D.N. Rao.
Professor D.N. Rao, good evening. Again, I express my gratitude for accepting our invitation to share the excitements of this year's Nobel Prize in Chemistry. And there is no need to introduce you because many a time you have spoken on this platform. Just a formality, I will tell you that he is the honorary professor at the Department of Biochemistry in Institute of Science. And he has extensive postdoctoral research in Switzerland, in US, and many other labs, reputed labs. His research for his focus is on restriction endonucleases and modifications. And he's also elected fellow of all the science academies of the country. And in spite of his busy lab work, administration, etc., he has devoted his precious time in terms of contributing to his thoughts, expertise to journals like Journal of Bioscience, Journal of Genetics, and Journal of Resonance. So this also adds to his commitment to the growth of science. And now I request Dr. Rao. Of course, I must again reiterate, he was the person who was responsible for my stay for 5 years at Institute of Science, sir. Thank you very much. I enjoyed your hospitality. And now I request D.N. Rao to enlighten us about CRISPR-Cas. Please, Dr. Rao. Dr. Rao, please. Unmute. Whatever.
Sir, please go ahead, sir. Make it full screen. Yes.
All right. Yeah, I'll make it full screen. 1 minute.
Yes, sir.
Okay. Is this clear now?
Yes, sir. Absolutely perfect, sir.
Okay, very good. What I'm going to tell you in the next 40-45 minutes is a beautiful story that was extremely unexpected and came right out of the blue. Genome editing and the breakthrough work that could change the world is probably the most appropriate title that I can think of for this year's Nobel Prize in Chemistry. And this year, it went to 2 women scientists, Emanuela Charpentier from the Max Planck Institute for Infection Biology in Berlin, in Germany, and Jennifer Doudna from the Department of Chemistry, the University of California in Berkeley, in the United States. And the citation for the Nobel Prize for Chemistry this year very clearly says, for the development of a method for genome editing. And I want you to note this is a method for genome editing. There are a number of other methods, but this is one method for which Emanuela Charpentier and Jennifer Doudna got the Nobel Prize in Chemistry. And these are the 2 individuals that I showed you at this time. I thought very quickly I'd spend 2 slides on the Nobel Prize in Chemistry. 110 prizes in chemistry have been awarded since 1901. And the most number of prizes in chemistry has been awarded to scientists working in biochemistry. And that is a more recent trend. And the first
Sir, sorry to interrupt, sir. Sir, sorry to interrupt. Voice is not clear, sir. Please check the mic.
Now, can you hear me?
Yes, sir. We can hear you. Yes, sir. Yes, sir.
Sorry. So the first Nobel Prize in Chemistry was given out in 1901 to Van 't Hoff. All of us read about Van 't Hoff's discovery of the laws of chemical dynamics and osmotic pressure in solutions. Marie Curie got 2 Nobel Prizes, one for physics and one for chemistry. And Linus Pauling is the other person who got 2 Nobel Prizes in chemistry and in peace. But Frederick Sanger is the only Nobel laureate who has received 2 Nobel Prizes in chemistry for the discovery of protein sequencing in 1958 and the nucleic acid sequencing in 1980. Marie Curie's family, known as the Nobel family, 5 members of the family, the same family have got the Nobel Prize. Marie Curie, Pierre Curie, Irene Curie, Frederick Joliot, and Eve Curie. But this is popularly called as the Nobel family. And I think the women getting the chemistry prize are very few. And I think there have been 7 or 8 now, or 7 actually. These are the first 4 that I have photographs of. This is the Curie, the two Marie Curie and daughter. This is Dorothy Hodgkin from Oxford, who got the prize for crystallography. This is Ada Yonath on extreme right, who got the Nobel Prize in chemistry for the discovery of for the structure of the X-ray structure of the ribosome. She shared the prize along with Venki Ramakrishnan a few years back. And 2 years back, Francis Arnold from the California Institute of Technology got the prize in chemistry. And this year we have two women now, Emmanuelle Charpenter on the left side and Jennifer Doudna on the right side for the chemistry prize. Now, before I actually give the details of what the prize is all about, I think I have to go back in history and emphasize a point here, especially for students, that working with prokaryotic system, working with bacteria and viruses has always paid off. About 65 years ago, a Swiss biochemist named Werner Arbor provided the first evidence for the existence of these nucleases of scissors that could cut DNA by doing some very, very simple microbiological assays. Experiments. He showed that every bacterium has a pair of enzymes, one that cuts DNA and one that modifies DNA, and this is called as the RM system, the restriction modification system. And this is actually a very primitive immune system. To go back in evolution, bacteria being the first organism to come. So a bacteria, it is living very happily. It has this RM system. But when the virus comes, a bacteriophage comes in, the bacteria don't like foreign DNA, and therefore they like to chop it off. And therefore the foreign DNA is actually chopped off by using this restriction enzyme, which is like a molecular scissors, and the foreign DNA is broken down to pieces so that the virus or the bacteriophage can multiply. But the pair of scissors can also cut its own DNA. The host DNA can also be cut by the same scissors. But that is prevented because the DNA is modified by putting these green circles here, which is a methyl group, and methylation of DNA protects the host organism from its own scissors. Whereas the foreign DNA in the form of phage DNA or viral DNA is not protected, is not methylated, and therefore it can be. And this is the discovery of RM system, and this is the discovery of restriction enzymes, and I don't have to tell you more about restriction enzymes. You can see how the whole area of recombinant DNA technology, the genetic engineering was all possible because of this fantastic discovery by the Swiss biochemist called Werner Arbor. Actually, the experiments that Werner Arbor did in the University of Geneva were actually done by his lab assistant called Daisy Boussois, whose work I'm going to tell you later on, and the unfortunate thing that has happened with Daisy Boussois. But the system of a primitive immune system, that is the restriction modification system, is actually an innate immune system. All of us have T cells, B cells, killer cells, and things like that. But bacteria, this is the immune system. Any foreign DNA will be chopped off by the restriction enzyme, and own DNA will be protected or methylated, and therefore these scissors cannot act. While this is 60 years back, 65 years back, this is the innate immunity. The CRISPR-Cas, the subject of this year's Nobel Prize in Chemistry, is actually again from bacteria and phages, and that is an example of adaptive immunity. You see, 60 years back, the first kind of immunity that was observed got the Nobel Prize. Now, again, this year, the other immunity system that bacteria have, called the adaptive immune system, has got the Nobel Prize. And this is where I think the discovery of many things in bacteria, in small organisms, actually the foundation of very large discoveries that are made in mammalian systems of eukaryotes. And what I want to tell the students is, what is true for the E. coli is also true for the elephant. Of course, you have some complexity, but the basic steps, the basic chemistry of all the reactions is common for E. coli, as simple as E. coli, bacterium, or the elephant, which is the very complex. So this is what I just talked about. This is the bacterial cell, and this is the host chromosome, which is protected by other group, which is methylated. And then foreign DNA comes in the form of virus DNA, viral DNA. It is chopped up into pieces, therefore it cannot survive. But you have now systems that people have discovered that the phages or the viruses have also become very clever. And they have now developed something called the anti-restriction systems. But we won't talk much about that. We move on to what these enzymes are all about. So this is
Sir,
what
sorry to interrupt, sir.
yes.
Sir, sorry to interrupt. The
Yeah.
voice is still it's breaking, sir. The modulation is not clear. Please check the mic, sir.
Yeah, I know. I'm checking the mic. Now is it okay?
We can hear you, sir, but in between it's breaking.
Yeah, I don't know how to control that. How do you control that mic? I've just put the speakers on. But that's a pity. Can you hear me now? I hopefully, yeah.
Yes, sir. We can hear you, sir.
Better, sir. Better.
All right. So Werner Arbel is a Swiss biochemist. He shared the Nobel Prize with two others from Johns Hopkins University, Dan Nathans and Hamilton Smith, for the discovery and the use of restriction enzymes. And I don't have to tell you the use of restriction enzymes because it is phenomenal. And everything that you can talk about genetic engineering or recombinant DNA technology is only possible because of the discovery of these enzymes. So restriction enzymes are enzymes naturally occurring in bacteria, and these bacteria use these enzymes to protect themselves from viruses. And these enzymes cut up DNA that the virus injects into the bacteria, so the virus cannot be reproduced. This is a good example. Here is EcoR1, which is a famous restriction enzyme, recognizes very specific sequence like this GAA, PTC, and makes a cut on both sides, and then you have two pieces of fragment and cut DNA. These are molecular systems. A similar kind of a nucleus is now associated with the CRISPR-Cas. That's why I brought up this particular nucleus chapter. Now, you can take a DNA, use a variety of restriction enzymes, and you get wherever there is a particular site, the site will be cleaved, and therefore you can generate a lot of fragments, which can be separated on agarose chains. Using another enzyme and a third enzyme, and by this you can map the whole genome and make a restriction map, which is shown. This is by simple use of restriction enzymes, and this is what is the basis for the Human Genome Project also. The sequencing projects, not only of the human, all genome sequencing projects, DNA has to be cut down into small pieces and then sequenced, and that cutting of DNA pieces is done by enzymes. Therefore, you see the potential of this discovery, which was made in the 1960s by Werner Arbel and others, and they were suitably rewarded with the Nobel Prize in 1978. So, restriction enzymes are, or restriction modification enzymes are just one part of this bacteriophage of our bacteria system. Now, usually, if you follow this carefully, a phage normally or a bacterial virus will absorb onto the bacterial surface. Then the phage will inject its DNA into the bacteria. The phage DNA is multiplied a number of times using the host machinery. Once the DNA of the phage is replicated, it's transcribed and makes proteins. And once the proteins are made, all phages are made, and then the phage kills the cell, and all the phages are released, and therefore you have a cycle that goes on. You must remember there are 100 or 1,000-fold more phages that we have in the world compared to bacteria. And we know that the bacterium in general is 10 to the 14 or 10 to the 15 number of bacteria, and you can imagine the phages are 10 to the 21 or much more than that. So phages have survived. Phages will always survive because they have a way of living in their host organism. In this case, it's the bacteria. So while RN system is just one of the measures of different systems, the CRISPR-Cas system, which the Nobel Prize was given this year, is another system. And that is what we're going to talk about, adaptive immune system. Is my voice okay, Raghunath?
Sir, I think if removing headphone may give the clear voice.
Maybe I'll try
Or you
it.
can try to reinsert that pin, sir.
1 minute. I'll try it. Now is it okay?
Yes, sir. Yes, sir. Now it's clear, sir.
Remove the headphones.
Yes, sir.
All right.
Yes, sir. Thank you, sir.
All right. Very briefly to tell you what is gene or genome editing. And this is a nice cartoon that I got from a friend of mine. This is a very German way of saying a thought experiment called the Gedank experiment. And the objective of this experiment, I've written that down, is targeted replacement of a mutant gene, which is shown in red color here, with a green one, or vice versa, in the human cell. And in the Godmas experiment, one could replace a normal gene with a mutant gene, and it essentially knocking out that gene in the cells. And this is how you find out whether what is the function of the gene. Because in many genes, we don't know what the functions are. If you knock it out and then ask what is the phenotype, then experiments are very limited. So this is what genome editing is. That either you replace the whole gene, or you mutate or you change one residue in the gene and see whether you can make a wild type to a mutant or a mutant to a wild type. And this is what is the genome engineering experiment that everybody wants to do. And that is what this particular technology called the CRISPR-Cas actually gives. So genome editing is a process of introducing specific modifications in the genomic DNA, which include deletions, point mutations, inversions, or translocations. It can be used for genetic correction. And why do you want to do genome editing? Is to understand the function of a gene or a protein. Because we don't know many proteins, many genes, we don't know the function. For example, about 20,000 protein coding genes in the human genome. We still have very little idea of what 6,000 of them or 7,000 of them actually do. So you knock out the gene and then ask the question, what happens to the organism? And therefore, reverse kind of experiments are very, very nice to be done. And genome editing can be done. But genome editing is not easy. You have to be very precise. And that is where the beauty of the CRISPR-Cas system comes. It's a very precise way of doing genome editing. But I must tell you that before the CRISPR-Cas was actually came into the picture, there were two other kinds of genome editing systems. One is called the zinc finger nucleases, and one is called the tailings. The tailings is the transcription activator like effector molecules. Now, I bring this because I think everybody thought that genome editing or genome engineering will get the Nobel Prize. And therefore, while CRISPR-Cas is just a method, there were earlier methods for 10, 15 years back before the CRISPR-Cas system that people, scientists actually discovered to do exact, precise genome editing. And I bring this point simply because one of an Indian scientist, Chandrasekhar Srinivasan, from Johns Hopkins University, was involved in the discovery of the first genome editing nucleases called the zinc finger nucleases. He also happens to be a very good friend of mine, and he spent about 9 months, 2 years back in my laboratory. What Chandra did was to take one of these restriction enzymes that I talked to you about, an enzyme that recognizes the sequence and cuts away from the sequence. So this is something called the example is the FOC1 enzyme. It recognizes the sequence, but doesn't cut within the sequence. It cuts away from the sequence. So the binding domain is one and cleavage domain is separate. And therefore, Chandra did some very nice experiments and actually made two domains, one called the recognition domain and one called the nucleus domain. So the nucleus domain alone, if you express it, it can cleave DNA non-specific. But when you fuse it with the recognition domain, it cuts very specific. So what Chandra did was to actually take the nucleus domain and add it with some other some other proteins which bind DNA, for which a lot of information was available from Karl Popper's lab. And he fused the DNA binding domain of this one protein with the nuclear domain of the FOC1 enzyme and made a hybrid enzyme, a designer nuclease. So you took a number of DNA binding proteins, added together. Each of these DNA binding proteins recognize very specific sequences which is shown here. And therefore, he created designer enzymes. He created enzymes that were not there in nature before at all. And because these DNA binding domains recognize specific sequences, he now was able to cut DNA wherever he wanted, that way you had these particular sequences. And these proteins require zinc, and therefore it is called as the zinc finger nucleases. And it's the first gene editing nuclease. This is Chandra here, it was in Chandra. And I think, Jennifer Doudna, in her book called A Crack in Creation, spends 2 or 3 pages describing Chandra's work, saying that these were the first genome editing nucleases. And I bring this point because he was from Pondicherry, he was a chemist. He did his master's in Madras and then went to Georgetown University in the US, and then did a postdoc in Johns Hopkins, and he's been there as a faculty member. And this was done in the 1991 92. This work was done. The CRISPR work came out in 2001. So many years gap, but the zinc finger nucleases are the first gene editing nucleases that ever went. I could go on about Chandra and the zinc finger nucleases, but I think this is an outstanding discovery, which unfortunately has missed the Nobel Prize. I think well, they chose to give a particular method and not the whole area of genome. Then there came another version of it, which was also prior to CRISPR-Cas, but this is from another organism called Xanthomonas bacteria, where they have a protein that binds, again DNA binding protein, and this also was used with FOC1 nuclease. And similar like the zinc finger nucleases, they also made a bit more specific than zinc finger nucleases. And the talents for the second generation of editing nucleases. And then came the CRISPR-Cas system. Now, the CRISPR-Cas system is very interesting and is the most popular method of genome editing because it's a very simple way of doing it. The zinc finger nucleases and the talents are slightly more complex, but we won't have time to elaborate on that. But this is what the CRISPR-Cas system is all about. And I have to tell you, some way back in 1994 or 1990, the mid-90s, I think, summer student in the University of Alicante in Spain was analyzing a lot of genome sequences. And you should remember that genome sequencing was a very tedious procedure. The automatic systems had not come. But in the literature, there were a lot of information available on genome sequences, and he was analyzing the genome of a very peculiar organism, Halophorix mediterraneae, which is seen in the Spanish, one of the coast of Spain. That he isolated the organism and he saw these sequences, and the sequences are here, which I repeat here. There are a lot of repeat sequences, and between 2 repeat sequences, there was a spacer, which was marked as 1, 2, 3. The repeat sequences were all palindromic in nature, that is just like the word Malayalam, because they repeated forward and backwards. And between 2 repeats was a region called a spacer, and this spacer sequences were all entirely different. This was part of the chromosome of this bacteria. And now, people have shown it in all bacteria that you have, many bacteria have this repeat sequence arrangement. And right after this sequence here, the repeat sequence and repeat spacer, you have certain genes called the Cas genes. And this is what Francisco Monita, who was a summer student, later on he became a faculty and called them as the clustered, regularly interspaced short palindromic proteins. And that is what CRISPR stands for. And the CRISPR associated proteins were called the Cas proteins, and the genes that coded were called the Cas genes. This whole thing, this element of the spacers, repeat, and the Cas genes are found in the chromosome of many bacteria. But even before Francisco discovered this in 1987, a Japanese scientist had observed this, and he just published a paper saying that there were repeat sequences. But the word CRISPR is given by this Francisco Mojito, and is attributed to the discovery of these sequences. Then he went around to find labs which would actually work on it. He actually spent some time in Jennifer Doudna's lab, and that is how this whole work progressed to a large extent, because a lot of biochemists, biophysicists were involved, and people were interested to find out what are these repeat sequences doing in bacterial genes. Now, to cut a long story short, and it's not a very long story, but since 1994, 87, this is what happened, and I want all of you to follow this. I have 3-4 versions of the same thing which I'm going to explain, so that you will get the gist of what the CRISPR-Cas technology is. Amnath, is it okay? Can you hear me?
Yes, sir.
Yes,
Yes, sir.
yes, please.
Okay. So when the bacteriophage actually infects, as you see from the top left, please follow my arrow here. When you see a bacteriophage absorbs through the bacterial membrane here, it injects its DNA. This DNA, the phage DNA or the viral DNA, is actually chopped off by 2 enzymes called the Cas2 and the Cas3. Remember the Cas genes that I talked about? And they're broken to small pieces. Foreign fragment acquired to the CRISPR locus, and these fragments are now put between the 2 repeats here. If this, the black one here is the repeat, between 2 repeats is this foreign DNA actually inserted. And therefore, this is called the CRISPR locus. The CRISPR locus are repeat sequences and phage DNA fragments. And next to it is the Cas genes, which are involved in the CRISPR technology, which I'll tell you. Just follow this. So the first thing is the DNA is injected, it's broken down to pieces, and these pieces are inserted in the bacterial chromosome between the 2 repeat sequences. Right? This is what happens. Now, the next time the phage infects the same cell, for example, in the meantime, all this is which is incorporated, this is all the DNA now, is transcribed to make an RNA molecule called the CRARN molecule, which is a combination. The DNA is being transcribed to form an RNA molecule. It's the same DNA with all the repeat and the spacer ones, which is transcribed to form an RNA molecule. This combines with another RNA molecule, a small RNA molecule called the transcoded RNA molecule called the tracer RNA at that time. This tracer RNA molecule and the enzyme called the Cas9, which comes from the genes of the Cas genes, combines with the CRISPR RNA, which is broken down into pieces to form a complex, and this Cas9 RNA complex formation now has this tracer RNA, it has the phage RNA, the DNA transcribed to RNA molecule, and becomes a potential enzyme, a very, very powerful enzyme, which is RNA guided endonuclease. And when this will keep on searching, and if it finds a foreign DNA like the phage DNA again, it will recognize that because it has complementary sequences within it, and that cut the DNA because the Cas9 is a nuclease, which then cuts it, and the foreign DNA is chopped off. That is what bacterial resistance because of the virus is attributed. So this is not like restriction modification, which is an innate immune system. This is an adaptive immune system because these pieces of DNA in the spacer are all acquired before. And the bacteria uses this to recognize again in the second infection. So it's a naturally occurring bacterial defense system. And this is a system that was exploited, and I'll tell you how it was exploited to give you this genome technology that has come up. So I'll go to the next slide, which is a repetition of this, but a different slide, just to show you again, and I want all of you to follow this. The viral DNA, the phage DNA, is incorporated in the spacers here, S1, S2, S3, between the repeat sequences. And then this whole DNA molecule here is transcribed. It makes something called the CRISPR RNA, which is broken down to pieces. So if this is the pre-breaking down, then you have smaller pieces of CRISPR RNA that's formed here. This molecule is combined with a trans-access coded RNA molecule, another RNA molecule, which has sequence similarity to this region of the DNA of the repeat sequence here, forms a complex with an enzyme called Cas9, and then it becomes an effector complex, and then this protein cuts the viral DNA, making it non-functional. Now, this is a system used by the bacteria to defend itself. It doesn't want phages, and therefore, if phages do get it, this is the way to kill the phages. So it's a defense mechanism. And I think, from here onwards, after this discovery, I think Jennifer Doudna actually crystallized on this whole phenomena and thought about a way of how to use to make corrections in the genome in a very precise manner. And that is what I did. So you must remember the layers in this CRISPR-Cas technology is actually the CRISPR RNA, the tracer RNA molecule, and the Cas9. These 1, 2, and 3 are the most important layers in this technology. While all this is made by the phage, now you can actually design an RNA molecule to a DNA molecule that you want to cut and combine this together to form something called a guide RNA. And with one RNA molecule, which has both the tracer RNA and the cRNA, you can add Cas9, you can add DNA exactly where you want the chopper, where the DNA to be cut. This whole complex will be cut very quickly. Okay. One more slide, just to make it very, very clear to you all. Why this injects the viral DNA? The viral DNA is put in the spacer between the repeat sequences here. This whole thing is called as the CRISPR DNA, which is now transcribed to make a long RNA molecule, which is chopped up by the enzyme called RNase3 to give you small pieces of this CRISPR RNA, which combines with the tracer RNA, another molecule. And this is where Emanuela Charpentier's work comes. She discovered this tracer RNA. And she found that this is how the RNA molecule is now, the Cas9 is guided to a site where you want, and then the chunks. So this is that very simple system. This is actually a slide by the Swedish Academy of Sciences, which gives the Nobel Prize, and it's very nicely written here. Actually, again, these are all slides just to show you the simple way that you can do this. Now, instead of the phage doing it, you actually synthesize an RNA molecule, the cRNA molecule, and the trans-activating RNA molecule, one RNA molecule, add the Cas9 and put the DNA where you want the cleavage to occur, and you will get the double-stranded DNA open up or broken, and you'll have anything that is possible in one sense. A lot of details here that these sequences right next to the repeat sequences or the protospacer adjacent proteins are all important. They are important for the Cas9 to the You have several Cas enzymes, but each of them have these sequences. This protospacer adjacent motif is the recognition motif. So the Cas9 is the protein which promotes genome editing by stimulating a double-strand break at the target gene. So conversion from understanding the bacterial adaptive system to making it as a single RNA molecule with Cas9 is the work of Jennifer Doudna, And for this, she has been awarded the Nobel Prize. Emmanuel Charpentier is the one who discovered this tracer RNA. She published a paper in 2011 and then went to a meeting in Puerto Rico in the US. And at that meeting, she met Jennifer Doudna. And together they worked on this whole system of doing genome editing, not in bacterial cells, but in non-bacterial cells, in mammalian cells, plant cells. And in 2012, they published a famous science paper, which is the paper titled by the Nobel Foundation for the discovery of adapting a bacterial immune system to genome. I think that is the greatness of this whole work. A very simple system exploited so that you can do lots and lots of very nice things. So the co-expression of the guide RNA, and the guide RNA is nothing but the tracer RNA and the CRISPR RNA and the Cas endonuclease enables the CRISPR-Cas9 genome. And this is the final sequence. So this is the PAM sequence, which is the important sequence which the Cas9 recognizes. The Cas9 has combined with the tracer RNA and the guide RNA, where hydrogen bonding between the target and RNA is made, and between the RNA and the main molecule, and therefore you have this whole system. And now this has been crystallized, and the structure of this whole complex. Now, what I want to mention here that the Cas9 creates a double-strand break in the genome, and therefore you can design a CRISPR RNA to target any sequence next to the PAM sequence, that is the recognition sequence for the Cas9, and you can actually go and edit wherever in the DNA. This is what Jennifer Doudna's and Emmanuel Charpentier's science paper said. That's where it opened up that any cell, not only bacterial cell, mammalian cell, plant cell, fungal cells, then all genomes can be actually targeted where editing is possible. So that is the greatness of this work that does come up. But I want all of you to remember that the CRISPR-Cas is just one method of genome editing. It's the best method, it's very efficient, a lot of advantages, whereas the earlier ones, the one by Chandra, the zinc finger nucleases were all discovered much before 1996. Another one called Cadence came out in 2010, and the science paper of Jennifer Doudna and Emmanuel Charpentier came in 2010. You can see the multiplexing, the targeting efficiency, and all the plus points of this CRISPR-Cas9 is most easily done, and anybody can do it. Once you have a guide RNA and you have Cas9, you can edit any genome. It's so simple. Now, there are some disadvantages of all these. There are many disadvantages of zinc finger nucleases and cadence. Even CRISPR, besides the target area that you want, there have been cases where there's off-target mutation. Therefore, a lot of care should be taken before you apply to human therapy and food sources, because cleavage of DNA at the wrong place can be detrimental. I wanted to tell you the story of the CRISPR. It started actually by Japanese scientists that who first saw the sequences, but didn't know what the sequences. And it was only in 2005 or 2002 the word CRISPR was, the name was defined, and Francisco Mojica identified the foreign origin of spacers and proposed the adaptive immune function. From there onwards, you go all the way 2008, 2009, 2010, 11. 11 is where Emmanuel Charpentier discovered the tracer RNA. And in 12, they actually both Jennifer Doudna and Emmanuel Charpentier published this famous paper in Science. But I think one has to remember some history, and there were many other players who also contributed largely to the discovery of this system. You see a variety of countries. It started with the University of Alicante in Spain here, and then moved to France and everywhere, all over the world, this technology has been used, or not been used, has been developed. And a number of groups have worked work on understanding. Just before the 2001 paper of Emanuela Charpentier and Jennifer Doudna, a Lithuanian scientist in Vilnius, in Russia, part of old Russia, Burgess Sixtus actually had the same mechanism, but his paper was published 3 or 4 months later after the science paper was published. And here's one person, Burgess, also should have been included in the Nobel Prize. But somehow the foundation decided to give only 2. Normally they give 3. This year they decided to give only 2. And Burgess Sixtus work, exactly the same work that Jennifer Doudna and Emanuela Charpentier did. He did independently in Lithuania. It went unrecognized. So why is there a craze of this for craze? Because simply because you can program the enzyme to perform genome editing and gene editing in mammalian cells, in human cells. And all that you require is an RNA sequence which can be synthesized, which is targeted towards the target sequence, and is simpler and easier than thin finger nuclease or TLNs, and an unprecedented efficiency and stunning ease of anybody can make very easily. And therefore, you can rectify bad genes, good genes, or find out what is functional gene and just do mutation like this one. If you look at from time the Jennifer Doudna's paper and Emanuela Charpentier's paper came out in 2012, you see now the number of global markets, a lot of companies, both Emanuela Charpentier and Jennifer Doudna have their own company, innovative diagnostics, which do all sorts of work on CRISPR-Cas. And you can see from 2017 itself, in billions, for example, global markets, what is estimated to be in 2025. So there's a huge rush because it's such an easy technology, and now you can manipulate genomes very easily. But there is a fear about it. If you look at publications, for example, on CRISPR, like 2000, 2002, hardly any publications. Now you see now there are 5,000 odd publications in 2020. Both publication and patents, because of the technology, have gone up so much that there is a patent war between 2 groups. Between Jennifer Doudna's group and another group on the East Coast, led by George Church and Feng Zhang at MIT Protein Institute, who have been fighting for this patent of the CRISPR technology. And I believe, as I know, the latest version and latest news is the Feng Zhang group in Boston have won the patent. The Nobel Prize went to Jennifer Doudna. So whether it be zinc finger nucleases or cadence or the CRISPR, you can do all this targeting very easily. Even the zinc finger nucleases, although they require more work, you can now target it a bit more specific, but ease at which the CRISPR-Cas system can be done is unbeatable. Here a good example. Your mushrooms, for example, if you just leave it in air, they become strong. But now by using CRISPR-Cas technology, you can make white mushrooms. You knock out the polyphenol oxidase enzyme here, and your mushrooms will never turn. You can make a white flour into a very nice, this is petunia, Japanese morning glory. You can change it from violet to white or white to violet. You can make cows without horns. There's a big industry in the US and the Western market to have cows without the horns. Or you can have nice example where there is one example which is the horn snail, which is right-handed versus, and you can make it left-handed. So handedness can be changed by doing just one mutation, one change. And this has been done by CRISPR-Cas. So you can imagine anything that you want to do with this, anything that you want to do, and CRISPR-Cas is a fantastic technology to do this changes the world. Here's another example of you have a patient with a blood disorder. Okay. So isolate the cells according to genome editing by either in cases or by the CRISPR Cas, change that mutation, make a lot of these things, and put it back and do a bone marrow transplantation, and you get back the patient. No more patients who have blood disorders which are rectified. So you can imagine now, and this is where all these companies which have come up now in recent times are doing this kind of, using this kind of approaches to set a lot of diseases back to normal conditions. You must also remember, I want you to remember that CRISPR-Cas technology is not just for gene editing. The system is a DNA detector, an RNA guide that can design one specific targets. That is what CRISPR-Cas9 technology. I'll give you one example. In the dairy industry, where a lot of milk and yogurts are made, a lot of phage infection kills the bacteria which are important for the dairy industry. And therefore, to kill the phage now, the dairy industry can use the CRISPR immune system to protect important bacteria like lactobacillus acidophilus. The yogurt manufacturers were actually ones who contributed a lot to understanding of this. Early in the 2000s, for example, a lot of research work was done by this company called Danisco, a Danish company which makes yogurt. It's now taken over. Another example is that it can detect nucleic acid, and therefore a kit made by Feng Zhang, a controversial guy. The other guy was also probably tipped for the Nobel Prize, but got the patent. He has actually developed a rapid detection of nucleic acid by using the CRISPR-Cas technology. He called it a Sherlock, named after Sherlock Holmes. And we in India at the Institute of Genomics, IGIB, CSR Institute, have now come with a cheaper version of detective called Feluda by using another Cas, not Cas9, but another Cas enzyme from another organism. And Feluda is an acronym for this Cas9 molecule. And it is named after Satyajit Ray's famed detective called Feluda. And it's cheap, much cheaper than the Sherlock and other detectors. And therefore, identifying COVID-19 virus has become very useful. This technology has come up in a big way, and has been, I think, is being used in India now. This is a technology developed by a CSR institute and the government. So you have a lot of things that has worked. The chemistry prize went to Emanuela Charpentier and Jennifer Doudna for the development of a method of genome editing, which was actually repurposed an ancient bacterial immune system. They've got lots of awards. Even bigger prizes than the Nobel Prize for the Kavli Prize, which is $3 million, was also given to Verghese Yanulaitis, Verghese Sixonus. These three guys shared it, but the Nobel Prize is given only to two of them. And therefore, one also talks about the ethical debate around this. Now that you can modify and you can manipulate genomes, especially human genomes, this has become a very big issue with this CRISPR technology. Alfred Nobel was actually condemned, because he was into explosives research, and he donated his money, and he said that I would support who during the preceding year shall have conferred the greatest benefit. CRISPR technology is a benefit to mankind, is a boon for mankind, but it can be misused. It can be misused to a wrong thing, and you want to have only certain kind of populations, therefore eliminate other kind of populations and things like that. So many important discoveries have complex social implications. An application not readily categorized as merely good or bad. How CRISPR should and might be used raises serious ethical and societal issues, and how far should we go? The minute Jennifer Doudna's science paper came out, a Chinese biologist called Yi Jiangqi modified twin embryos, resulting in the birth of two girls allegedly containing alleles that would confer protection from the infection by HIV. So it worked with the human cells. But since he had not taken permission and did not follow regulatory rules and things like that, he was actually arrested and put in jail. He's still in jail. So those regulatory mechanisms of whether we should tamper with genome, our genome, is a very big issue. Only when you're terminally ill and things like that, gene therapy must be done. And therefore, Jennifer Doudna, in a book, she has actually said that genome editing of embryos should be possible only where there is a clear unmet medical need. Is there? One has to be very careful about this technology. The science is not divorced from society, and its goals and implications are not a neutral competition. And that is where I think the Nobel Prize highlights this also. Who gets credit for CRISPR? Not the prestigious award to all the people that I mentioned, but finally it is Emmanuel Charpentier and Burgess Sixinus from Vilnius in Lithuania. Mr. Prize George Church from Harvard. This is Feng Zhang from the Broad Institute at MIT. Many people, these are people who have contributed a lot to genome. So I think, whole genome editing started off with Chandrasekhar. But because the technology was not so easy as the CRISPR technology, we actually passed. But surprisingly this year, if you read, there's a lot of things in there saying that the first author on these papers of Jennifer Doudna or Emmanuel Charpentier, the students and the postdocs, they should also be recognized. I think the soaring popularity of gene editing has made celebrities and the principal investigator who pioneered the field. But the graduate students and postdocs are often. So there's a lot of debate on this, a lot of discussion, not debate, that the first authors of these papers, students who actually contributed to the work, will also be. One of the last slides, I have a couple of last slides here. This is Jennifer Doudna with a lab assistant. 20 years back, this person, Kai Hong Su, was with her, and Jennifer Doudna says, the laboratory success to someone she took a chance on 20 years ago, a laboratory manager. Anything good, anything bad, God, I think everything worked very well in the lab because she managed it. So it's a lab assistant and we do a lot of these things. This is the book that I would like many of you to read. I just got this book. This is written by Jennifer Doudna in 2015 or 16, much before the Nobel Prize. It's called A Crack in Creation, Gene Editing and the Unthinkable Power to Control Evolution. Because you can manipulate the place that you want, wherever you want. I think this is the power of this. So the Nobel Prize in Chemistry honors work for the development of a method for genome editing, which is all very nice. But I think, many chemists are very bothered because they think the chemistry Nobel has been biologized again. It's sometimes hard to see how an enzyme or a phage is chemistry, but they are. It is chemistry that governs the shape of these molecules, which in turn affects the function. And what last year, 2 years back, Francis Arnaud said, chemists are inspired and awed by the biological world. But what they should really be inspired by is the process by which biology discovers chemistry. That is called evolution. I think everything in terms of evolution becomes, and I think Ramadan will certainly agree on this, that biologists always do this. So it is the principles of biology have profound implications for technology and medicine. And as you see, a defense system in bacteria has now been exploited to give you this beautiful technology which is used in medicine. And this is my own thing that funding agencies always should fund basic biomedical research. And interdisciplinary research is what is the name of the game now. Therefore, I think invest in long-term blue sky science, in people, in ideas and teams. Take the risk of infrastructure and let people follow their ideas. This is exactly what happened to Jennifer Doudna and Emmanuel Charpentier. So my last slide, and I go back to evolution way back. Way back when life started, bacteria were very happy. You can see the smile on them. Because whenever pages came, they were killed and therefore pages were very happy. But as time progressed, millions and millions of years, as evolution progressed, the bacteria have developed anti-restriction system, now the CRISPR system. Now bacteria are happy. So there's a constant arms race between bacteria and phages. And I think studying these systems is very, very fascinating. And a lot of information has come out. Technology has come out, and you see. And I think I will end with this, that students especially nowadays are all fascinated by gene therapy and cancer and things like that. It's all very exciting. But studying simple systems like a bacteria phage can reveal more exciting things and more meaningful things. I'll stop here and thank you very much.
Thank you very much.
Mr. Rao, thank you so much. From all of us, you have taken us for a journey of the last 3 decades. For many of us, have been hearing about CRISPR, reading about CRISPR. Today, you have given us a hands-on experience, absolutely. And also, all the applications and the dimensions that you have brought in were so very interesting. And the future of CRISPR, how it might be taken through all over the world, is also something very interesting that you brought in your talk. We are so very grateful to you, Professor Rao, for this kind gesture of joining us in this endeavor. Would request Professor Ranganath for his remarks.
Thank you, sir. There's no remark. Just to say heartfelt thanks to both Soumitra Das and Dr. Rao. Last couple of slides reassures me that evolutionary biology, importance of evolutionary biology, nothing makes sense now. Therefore, whatever we do, we'll come back to interpretations and interrelationships among organisms. And great, sir. Again, both of you today, Rao and Das, again and again, it showed us how critical thinking is important in science. Critical thinking, critical teaching, and curiosity-driven research. And both the 2 Nobel Laureates of the chemistry today, they started their journey with their curiosity-driven research in their lab. That's how, see, people never think that there is immune system in bacteria. Always we think of immune system in human beings, but great story, great message, and unifying principles. Thank you very much, sir. Academy has started this new, and it is beautiful inauguration both of you. And all of us, Soumitra Das and Dr. Rao, I'm extremely grateful to you. Thank you.
Thank you. Thank you.
Thank you, Professor Ranganath, for bringing these 2 eminent in this endeavor of webinar on Nobel Prize Lecture Series 2020. To Professor Ranganath, thanks to Professor Dr. Professor Das from Calcutta, Professor Rao from Bangalore. To you, grateful. We are grateful for the Academy. My colleague, Dr. A.M. Ramesh, Chief Executive Officer, Academy would now speak. We are also grateful to our Member Secretary, Dr. Sobarad, who had been with us, and to all the eminent other colleagues who are there along with us, but you might be seeing on the screen, and the audience who are with us. Over to Dr. Ramesh, please.
Wonderful talks, enlightened very much. We take this opportunity to thank immensely first Professor Soumitra Das sir, who gave a wonderful talk on Nobel Prize in Physiology. Then followed by that, Professor D.N. Rao sir, who gave a wonderful talk on Chemistry Nobel Prize. In fact, Professor D.N. Rao sir had given a webinar talk last month also on CRISPR technology. This talk was also excellent, like the previous one. I would like to thank both of you immensely again from KSTA. KSTA will be organizing many webinars and workshops regularly. We will be requesting you for many more talks like this in coming days. Thank you very much, sir.
Thank you.
I would like to thank our coordinator, and honorable member of KSTA, Professor Rangnath sir, who initiated this new Nobel Lecture Series program. We are very grateful to you, sir, for this wonderful program. Thank you, sir. I take this opportunity to thank our Honorable Chairman, Professor Ayyappan sir, for all his encouragement in organizing this program. I also thank our Member Secretary, Professor Dr. Prakash Subrat, for his support in organizing this program. And many participants have come, not only from India and also from abroad. There are participants from 9 countries, right from Greece, Ecuador, Philippines, Montana, Sri Lanka, Uzbekistan, Malaysia, Indonesia, and Bhutan. Truly, it is an international webinar. We thank all of you for making it a very good event. I also thank senior participants, including senior academicians like Professor S.R. Niranjan, who has been there with us for this webinar, and all the teaching faculty, research scholars, students who have been watching this webinar, and also my colleague, Mr. Umesh, for making this webinar a successful one. Thank you very much. We will meet again with a new webinar. Thank you.
Thank you.
Thank you very much.
Bye, Soumitra.
Bye.
Thank you, Rao.